Good morning, students. Welcome to the biology class. So far what we have learned is that amino acids are the basic building block or the component of all the proteins, and we learned the structure of the amino acid. We learned that they are amphoteric in nature. Amphoteric means they can react with both alkali as well as with the acids. And the next thing we learned is that the zwitterionic form, in which a neutrally appearing amino acid would be carrying equal charges of positive and negative. Such a situation of carrying both the charges at the same time is called as the zwitterionic form of a biomolecule. Example, amino acid is a good example to represent this phenomenon. Now we learned that there are some amino acids which are sweet also, and then we learned that amino acids are more than 200, but in proteins only 20 amino acids are found, and we call that number of 20 as the magic 20. Then I gave you a very easy acronym to remember. How do you remember? Neutral, then the acidic, then basic, then sulfur containing, alcohol containing, aromatic ring containing, heterocyclic ring containing, and the amino group. So we learned all these things in the last class, and then we learned that there are non-protein amino acids also which occur freely, which occur in combined state, which occur in the non-protein substances, and we learned three examples from the human body itself, that ornithine and citrulline are the two amino acids which are non-protein amino acids, and they are very crucial in the urea cycle. Urea cycle occurs in liver, in which ammonia reacts with carbon dioxide to form urea. So in this process, these two amino acids play a crucial role. Then we learned about a neurotransmitter which has an inhibitory effect, and it performs the effect with the help of opening of the chloride channels, and the name of the amino acid is gamma aminobutyric acid, or the acronym is GABA, GABA. Then we learned about the derivatives of amino acids, that amino acids can undergo changes, that means they are reactive in nature. They may undergo methylation or acetylation, they may undergo phosphorylation, they may even undergo oxidation or the joining of the hydroxyl group. They can even develop disulfide bonds, specifically in the case of cysteine, because cysteine and methionine are the only two amino acids that carry sulfur. So cysteine is able to, whenever two cysteines face each other in a chain, that means they come closer, they have the tendency to develop disulfide bond. This bond develops between sulfur of one amino acid, that means cysteine, and sulfur of the next amino acid, that means the cysteine. So disulfide bonds provide stability to the structure of proteins. Then we learned that peptide bond is the bond which helps in joining two successive or two adjacent amino acids, and this bond develops between N and C and is represented as NHCO bond. In this process, a molecule of water is lost. Then we learned when two amino acids join together, it is called as dipeptide. When equal to or less than 10 amino acids are there, it is called oligosaccharide, and if it is more than 20, it is called as the polysaccharide. We learned that proteins are called proteins because when scientists found the diversity which exists in the case of protein, they found it to be the maximum. That's why they started calling it as the most important or the first one in rank. That's why the name introduced is protein. Remember, we learned in the last class that proteins are not homopolymer like glucose joins to form starch, glycogen, or fructose joins to form inulin. In the case of protein, there are 20 amino acids, so there will be hardly any protein which is homopolymer. They all are heteropolymers in nature. That means they are made up of different amino acids. And when we see the cell content, the dry mass of every cell on an average would be 50% protein. That means protein is the most abundant organic molecule inside a cell. And then we learned that proteins are of two types according to whether they carry essential or non-essential. Let me quickly revise. Essential amino acids are those amino acids which are basically animal proteins. The reason is essential amino acids are those which human body is not able to synthesize. So we learned that in the case of human, there are 8 essential amino acids, And because human body is not able to synthesize them, then we need to take them every day in our daily diet. And then there are two semi-essential amino acids which human body is able to synthesize, but the rate of synthesis is less, and the rate of uptake, that means use, is more. So total, that means there are 8 plus 2 essential and semi-essential amino acids. So if a protein provides us all these essential and non-essential semi-essential amino acids in the daily dose correctly, it is called as a first class protein or a complete protein. So all animal proteins are first class proteins, but when we talk about plant proteins, they are deficient in one or two of the essential or the semi-essential amino acids. That's why they are called as second class or incomplete protein. And the next thing is, that's why those who are basically vegetarian, strictly vegetarian in their diet, they are suggested always by the dietitian that they should go for more types of proteins like cereals and all. Otherwise, soya bean is the only exceptional plant protein which provide us all the essential and semi-essential amino acids. Then we started this when I told you from this topic we are going to start today. So the topic is regarding how many chains are there. We have already learned amino acids are formed by joining of proteins are formed by joining of amino acids to form a long chain. Now, how many chains are there in a protein? Accordingly, proteins are called as monomeric proteins or otherwise oligomeric proteins. So those proteins which are made up of only one chain are called, one chain means one polypeptide chain, are called as the monomeric protein, and the protein which is made up of more than one chain would be called, between two chains or more than two chains would be called as oligomeric, or the other name is multimeric. Please remember, there is no polymeric in this concept, only monomeric is there. Only monomeric is there and oligomeric is there. We don't have any concept of polymeric proteins. So either it is made up of only one chain or more than one chain, that means two chains or more. It won't be hundreds of chains. That's why there is no concept of polymeric